Welcome to NOAA Live Alaska. My name is Lisa Hiruki-Raring, and I'm going to be moderating today's webinar. This series is a collaborative effort by NOAA Fisheries Alaska Fisheries Science Center, where I work, NOAA's Alaska Regional Collaboration Network, and NOAA's National Weather Service. I'm also working with my co-coordinators, Story Oates and Mabel Baldwin-Schaefer, who will be helping me behind the scenes. This webinar series is designed to help you get to know NOAA's work in Alaska, our partners, and the communities we work with, and how we connect with your community. NOAA, the National Oceanic and Atmospheric Administration, studies the ocean and the atmosphere and where the two meet, from weather to ocean to the animals that live around us. This school year, we're presenting monthly webinars that feature our partnerships with organizations across Alaska. We're aiming to present perspectives from the communities we work with, as well as research conducted by established scientists and students. Today, we're introducing you to Bill Carter, a fish biologist with the U.S. Fish and Wildlife Service at the Selawik National Wildlife Refuge. While we'll be talking about NOAA's and partners' role in research and stewardship, we want to recognize that we're all coming to you from the traditional lands of Indigenous communities who have substantial traditional and local Indigenous knowledge and much to share with us. Bill is presenting from Kikiktagook or Kotzebue, where he lives and works. He works in the Kotzebue Sound area, which is home to several tribes of Inupiat people, and does field work on the Selawik River, which is the home of the Silmi Inupiat. We're grateful to the Inupiat who have stewarded their lands and waters for thousands of years and are indebted to them for their wisdom and knowledge of their lands and waters. We would also like to acknowledge that we're hosting this webinar from the traditional lands of the Coast Salish, including the first people of Seattle, the Duwamish people past and present. Story is joining us from Juneau, Alaska, the ancestral homelands of the Aquan and Takakwang Tlingit people, and Mabel is joining us from Benta or Wasilla, the ancestral homelands of the Dena'ina and Atna peoples. The community thrives thanks to the continued sharing of vision, wisdom, values, and leadership of these Indigenous communities. A few guidelines before I hand you over to our speaker. You're all muted because we have a lot of people on the line and we want to make sure everyone can hear our speakers. However, there's a box where you can write questions and we encourage you to ask them as we go. And my colleagues Story and Mabel and I will be keeping track for Bill. He'll stop every now and again and answer a few questions. We may not get to all of our questions, but we'll try to answer as many as we can. Okay, I'll hand it over to Bill to introduce himself.
Hi. As Lisa said, I'm Bill Carter, and I work for the U.S. Fish and Wildlife Service. I'm going to start the presentation here.
I think you just need to advance your slide, Bill.
Okay. All right, so we're going to talk about how fisheries biologists track fish throughout their lives. First, I'm going to do a brief introduction of how I got here and where I work, and then we're going to talk about current tracking, and then we're going to talk about how fish biologists figure out where fish have been before we were able to get our hands on them. And we'll be stopping a lot for questions. So I grew up here and moved to New Smyrna Beach in Florida when I was 7 years old, and I spent a lot of time in the tidal rivers and beach, and was a lifeguard throughout high school and college. And I went to college way up north in Ashland, Wisconsin, and the outdoor ed program there gave me an opportunity to try some new water sports. My first job out of college was actually working for NOAA as a fisheries observer in Dutch Harbor. And this job, you're out on a boat with the fishermen and you're looking at the catch and the bycatch. Sometimes the bycatch is really odd. This guy was feeding out of the net, and after he had a little rest on the trawler deck, he went and hopped off that railing off the back about 30 feet into the water. So after a couple of tours of that, I went back to Ashland looking for a job in my field, fisheries, and ended up doing a lot of other jobs, not finding a fish job. But I did find a new sport. I started dog sledding there, and that gave me an opportunity to come back to Alaska, where I ended up working for several Iditarod mushers as a handler. And then I worked during the summer in Denali National Park, and then started working with the U.S. Fish and Wildlife Service in the Fairbanks field office as a fisheries technician that took me all over the state doing all kinds of fish work. And then in 2014, I moved to Kikiktoruk, or Kotzebue, and Kikiktoruk means almost an island. So not only is the little sand spit we live on almost an island, but the Baldwin Peninsula is almost an island also. So this is an outline of the Selawik National Wildlife Refuge. It's about 5,000 square miles, or 3.2 million acres. It's larger than the state of Delaware and Rhode Island combined, and almost as big as the state of Connecticut. And the refuge straddles the Arctic Circle. So I live about 30 miles above the Arctic Circle. So that was really quick, so we can get to our other, the main topic here. And if you have any questions, you can type those in, and then we'll start moving on to the tracking part.
So Bill, you got one question of whether you use any of your lifeguarding skills up in Alaska.
I actually do, but I don't get in the water as much as I do when I was in Florida. But just all the water sports that I did in Florida help me when I'm in boats or out kayaking here, and just the general being aware of situations that might get people into trouble, because I'm often very far out in the field with a group of people of 5 or 10 to maybe as many as 20, and my lifeguarding radar is always on when I'm out there. So I'm looking for things that might get us into trouble, because it's not easy to get out of there if somebody gets hurt.
Right, right. Yeah, I'd imagine that would be so. Michelle was wondering what you did at Denali.
I actually worked in the tourist industry. I was a driver my first summer on the east end of the park, what they call Glitter Gulch, and then I worked for the Kantishna Roadhouse, which is one of the big lodges at the west end of the park, as a naturalist guide. So I took people out and showed them lots of wildflowers and everything else that might be out there at the time. So a few bears, a few caribou, some moose.
We did get another question about, through all of the different jobs that you've done, do you find that you have that each there are skills from each job that you take to your next job and that are useful?
Oh, absolutely, absolutely. Yeah, like I said, when I spent a lot of time in the outdoor ed department at Northland College, because I learned a lot of outdoor skills, which I have used. I was in a whitewater kayaking class that taught me how to move like boats that are sunk and things with ropes, and I've used that to get boats unstuck, or I've used that to haul gear into the river or out of the river. If it's full of water, it's really heavy, so being able to get some mechanical advantage has really been helpful. So yeah, I always pick up a little bit here and there and move that on to your knowledge set.
And then we also got a question about, Katie asked, what is your favorite kind of fish to study?
Well, it's kind of hard to pick a favorite, because there are a lot of different things that do a lot of really cool things. I like fish in general because they've been on the planet for a really, really, really long time. So they do all kinds of really interesting things. They can change sex. They can live in very different waters, like anadromous fish can go back and forth between freshwater and saltwater, and the fish that we're going to talk about today can go between freshwater and saltwater and back to freshwater and back to saltwater. So they've been around a long time to evolve into a whole variety of different things. There are more fish on the planet, different species of fish on the planet than there are birds, and the only group that has more species are arthropods, so insects and crabs and lobster, things like that. So
my mistake, I misread the name. It was Katya who asked that. And actually we have a question from Paul T. Albert Memorial School asking what's the most unique fish you've seen or studied recently. And I don't know whether that will lead into your next section, but I thought I'd ask that question as well.
Yeah, so well, the fish that I study are we'll talk about the groups here in a couple minutes, but they're interesting mostly because nobody else has really studied them. So every time I go out and I usually find something different, but I'm also a scuba diver, so marine reef fish are really cool to me because they do all those weird things like change sex and have weird living habitat or habits and things like that. So yeah, I really like all the fishes.
Well, it's a good thing that you're a fish biologist there. We have one more question before maybe we should move on to your next section, but there was a question about of the villages that are in your area. So I see you have Kayana and Norvik and Selawik highlighted there. How large are those villages? How many people?
So Selawik has about 800, and all the other ones I didn't. This map is from a different presentation, so I didn't put Ambler and Shugnak and Kobuk, which are all on the Kobuk River, just north of the refuge there. But all the other villages are between 3 and 5, maybe 2 and 500 people. So Kotzebue is the biggest. We're about 3,200, 3,500, something like that. And then Selawik's the next at 800.
Great, thank you. All right, well, maybe we'll hold the rest of our questions and we'll let you get on to your next section.
Okay. So we're going to use this picture to ask a couple of questions first. So what do you think makes tracking fish difficult? And this picture is a normal wildlife picture, but what do you guys think might make it difficult to track fish?
Right, so for those of you who are in our audience and classrooms, maybe you can put together some questions, some answers for your teacher to type in. What do you think makes tracking fish, figuring out where fish go, difficult? And you can just type in any ideas that you have into the question box, and we'll read out the answers as they come in. So Miss Ravenscroft's elementary school class from Akutan is saying that maybe you would put a tag on a fish. And Brenda is saying they move fast. Ms. Kate's 5th through 7th graders from Prince of Wales Islands is saying that seals and bigger animals eat them, so that might be a difficulty in tracking them. Let's see, Port Alexander School is saying there's so much water that it's hard to track them through that water. Michelle is saying that there's no way to communicate with them, maybe because they're in the water. And Melissa is saying there's similar appearance of fish, so it's hard to tell them apart. There's predators, so the fish or other animals that eat them. You have to be underwater to track them unless they're tagged, so that could be a difficulty. Miss Ryan's class is saying you can't see them underwater. The Miss Munier's class is saying, can you please allow more quiet thinking time? We suggested that weather, constant movement, and predators might be an issue. And let's see, some other ideas that are coming in are maybe the loud noise of approaching people scares them off. Igig School says that it must be difficult to put the tracking devices on them, and there are many animals that eat fish. And also Kenzie is saying, how hard is it to track fish? And so that is one of the, maybe this is one of the things that you could answer is, how hard is it to track fish, and what things make tracking fish difficult?
Yeah, so those are all good answers, good ideas. So the main thing that makes tracking fish difficult is, if you look at this picture, we breathe air, caribou breathe air, seals breathe air. So when they're in the air, it's easy to see them. So and fish being underwater, hard to see them. So you can't just take the binoculars out and count the 34 plus caribou that are running by there, and you can't count the 18 seals that are in the water here, but there may be more seals in the water because some of them might be diving. So you'd have to mark those seals somehow so that you knew between which ones have been counted and which ones haven't. So one more question. Anybody can think of some tools that we could use to figure out where fish move? And I know.
So the question is, what tools can we use to find out where fish move? And we had gotten a suggestion to maybe have some quiet thinking time. So maybe that's one of the things that I will wait for a few seconds, and then I will start reading out some of the answers that are given. And of course, some of the things are things that you've already talked about a little bit in the answers to the previous question. So having given a couple of seconds of quiet thinking time, let's see. We have, okay, so some of the suggestions that are coming out as tools that we can use to find out where fish move. Ms. Ryan's class is saying tracking. Michelle is saying tags. Ms. Raven's class, Croft's class is saying radar, tagging. And Ms. Cadahay's class in cake is saying by using food to get them where you want them. So like bait or something. Iguyig School is saying underwater cameras, GPS, tagging, our eyes. Ms. Kate's class is saying GPS tracker, tags, satellite pictures. Ms. Anderson's class is saying GPS, radar, tracking devices, look at fish scales with a microscope. I can see that some of these folks have looked at have studied fish before, because we'll be talking with that a little bit more. Ms. Doherty's class is saying sensors in the water. Port Alexander School is saying tags. Let's see. There's also fish bait, net, tags, binoculars for water like on a submarine. Ms. Cruz's class is, oh, these are questions, so I will ask those a little bit later. Ms. Doherty's class is saying study the currents and genetic markers. So are these some of the tools that you're thinking about, Bill?
Yes, there are a whole lot of good ideas there. And some are really applicable to different size fish or different environments. So yeah, we're going to talk about two here coming up. So like I said, there's about 34 caribou. There's a big bunch here in the center of the group that are, let me see if my marker is going to go. So there's a big group here in the middle that, let me get my screen on the right screen here. There we go. This big group in the middle is kind of hard to count, but I counted 34 in here, and there's 18 heads of spotted seals in that group there.
That's a lot of animals.
Yeah, but what you don't see, having trouble with my, What you don't see is the hundreds of thousands of rainbow smelt that those seals are feeding on. So in this situation, this was right outside of Kikiktorok here. We had a big open water, and we didn't even know that there was a smelt run at this time because it's usually under the ice. And this was just happened to be a winter where there was no ice in front, and the seals could come in. So then when we started looking, oh, there are all these seals here. Why are there seals here? And we threw out a net and caught tons of rainbow smelt, and they were all ready to spawn. So these are the main fish that I work on. They're related to salmon. They're commonly known as whitefish. And here you see what's considered the upriver or inland Inupiaq names for them. And I worked on my master's degree on the Cruchila, which is the broad whitefish. And most of what I work on now is sheefish or sea. And they get really big here. So they can weigh 50 pounds and be 4 feet long. And the main way we catch them is under the ice in the spring, just starting to do fishing this time of year. And they're so important here that several kids or high school students have done science fair projects on them. And that's what's going on in this upper right-hand corner here. They were looking at the commercial catch of sheefish and doing a genetics study and looking at their what they were feeding on at that time of year. So when I asked you what kind of tools we could use, lots of different types of tags came up. And first I want to mention this pop-up satellite tag and the website that's listed at the bottom here, gttops.org. We don't use these pop-up satellite tags because they're kind of too big for sheefish, but that website is really cool because it shows you all kinds of different animals, from sea turtles to marlin to tuna, all kinds of different things, and how we can track them with these satellite tags all over the ocean. So in the center there, you see 3 tags. The one on the far right, the green tag there, is what we call a T-bar tag, kind of self-explanatory. It's the same kind of tag that you'll see on your clothing, except it has a unique identifying number, who tagged it, and what agency tagged it, and then a phone number. And in the bottom right there, you'll see that we just poke it in behind their or between their fin rays, and it stays there for years. We last tagged fish with this type of tag in 2005 in the Selawik River, and we still get tags returned to us from fish that people have caught. So almost 20 years now, and those tags are just still hanging around on these fish. So these tags are used a lot when we don't know very much about fish, because they're super cheap and we can put out thousands of them. And then that way we can figure out where we want to concentrate our efforts and use these other tags, which are much more expensive. So the center tag there is what's called an acoustic tag. And we'll discuss how each one of these works in the next slide here. But the acoustic tags and the radio tags are both surgically implanted. And down in the right, bottom left there, one of our local guys, Sonny Berry, he's putting in an acoustic tag, just a little slit, pop it in, stitch it up, just 2 stitches and it's all done. The fish recovers really quickly because it only takes about 5 minutes to put one of these tags in. And then my colleague Teresa there is putting in one of these big radio tags, and we have to use a long needle to thread that antenna out because the antenna has to be outside the body. So it takes a little bit longer to do, but fish are under anesthesia, so they're asleep during this, so they don't feel much, and they recover really well. And The radio tag in this picture, because of the programming, we can make it last for 7 years. So they're in there for a long time, and they're really helpful. So radio tags are really good because they can be tracked over long distances. And this map that I have up here is from my master's thesis, and it shows the stationary tower array that we used, and we use this for several different fish. Several different fish species, and I tagged my fish here in the middle of the Yukon above the Tanana River, and they went way up here to spawn, and then they all went way back down here to the lower river. And the problem with radio tags is when they get down here to the lower river, where the salt water mixes with the fresh water, radio tags don't work really well. But when they're up here in the fresh water, anywhere up in this area, we can tag, or we can track them with a lot of different methods. We can use a boat, we can use snow machines, and we can even use airplanes. So we can track over a really large distance in a short amount of time. And then acoustic tags are really helpful because they don't have the same limitation as radio tags as far as what type of water they're in. So this is a map of the Kobuk Lake or Hotham Inlet, and then Selawik Lake down here. And we put out an array of what are called hydrophones, which are underwater microphones. And if you have at least 2 of these, you can get a very precise location, not just in relation to the hydrophones, but also depth in water, because there's only 2 spheres where those 2 signals will cross. So it's really a precise method of figuring out where a fish is. The downside is you can't use an airplane or even a lot of boats because they're loud if you're going at any kind of speed, or there are a lot of waves, which means you have to have a wide array of hydrophones out there, which are kind of expensive. So so another question, what kind of places are important to find?
Okay, so for our audience, we've got a couple of questions that are adding up, but I think we'll be pausing for questions shortly after this slide. So Bill's asking the question, what kind of places do you think that it's important to find when you're tracking fish? Like maybe thinking about what kind of things you want to find out about the fish. What kind of questions would you want to ask of them? Miss Ravenscroft's class is saying that you would want to find out where they lay their eggs. And a couple of other folks are talking about other places that are important to find. So Michelle is also saying where they spawn. So that's another way of saying where they lay their eggs. Miss Kate's class is saying that you want to find out where their food is, so what the food source location. Let's see, what else? So thinking about that map that you showed where they were traveling great distances, I suppose one other question would be how far upstream that they go. And actually Miss Ryan's class, we're just seeing that, where they travel, how far do they go? So Port Alexander is saying where they spawn as well. And Tununak, sorry, I mispronounced your school name, is you want to find out undercover in water, near the bottom, shallow, near food. Those kind of places might be important to find. Miss Katahe's class and Cake are saying that you want to find out where they find food. And Kristen is saying you'd want to know the type of water content and the temperature that the fish can be in. So what do you think, Bill? What are your aims when you're looking for where fish go?
Those are all great answers, and somebody mentioned water temperature and things. So some of these radio tags we can actually get that kind of information from water temperature and light and things like that. They're a little more complicated to put in, but yeah, we can find some different things. But yeah, all those suggestions are really exactly what we're looking for. So spawning areas. So here's some brown trout, and they're making a red. We have some bluegills, a white spotted pufferfish. That's one of the cool things that I really like, is like a little pufferfish made this really elaborate nest. But like these bumphead parrotfish that do what's called broadcast spawning. All the whitefish are broadcast spawners, so they swim upriver, get in a big group, and then release their sperm and eggs all together, and it mixes together and then drifts downstream and finds little cracks and crevices in between the gravel. All their eggs are really tiny. They're about the size of a pin. And the other place, like people said, are feeding areas. So where do fish find their food? All right, more question times. So
All right, we've got we've got a bunch of questions that are all lined up here. So I think that you had talked about what you use to insert the tags into the fish. Katya was wondering, do you use a needle to insert tags into the fish?
There are some kind of tags that you can just use a really big needle. They're called pit tags, and they're usually used for really small fish that are going through really small streams, like juvenile fish moving around. But for the most part, a tag that's going to last very long is going to have a surgical implant, and there's different types of surgical implants. Like the acoustic tags don't need anything sticking out of them, but radio tags usually do. So yeah, there are a lot of different techniques depending on, and like those pop-up tags just work very similarly to the T-bar tag. They just get poked in underneath the or between the fin rays, and then there's a link that rusts and it breaks off and it floats to the surface. And they actually use similar satellite tags for seals and things, and they actually just glue them to their heads. So every time they come out of the water, they can broadcast to the satellite.
Great. So Peter from Ms. Cruz's class was wondering, what kind of hook do you use to catch the fish?
Oh, that all depends. So sheefish, we use a big metal spoon usually, sometimes some swim baits that look like fish. They are fish-eating fish or piscivores, so we use something that looks like a fish.
And then Kristen and Katya were wondering, what is the biggest whitefish that you ever caught?
Oh, it's probably, the biggest whitefish is a sheefish, and it's probably in the 30-pound range, 3 feet or more long, something like that.
That's pretty big. I can't even imagine.
Not as big as the one that I showed at the beginning there, my friend Alex.
Right. So Miss Kate's class was wondering, why place the tag in the stomach of the fish instead of down the throat? Wouldn't they be more likely to die if you have to cut them open?
One, fish are really tough. Because I handle so many fish, I've seen fish with just some really nasty scars that they have survived without any issue. Well, maybe not without any issue, but they've healed from with and are back spawning without any problem there. We are really careful with the fish. We do just all kinds of things. We anesthetize them. We use disinfected water to rinse them when they're in the when we're doing our surgeries, and we try to do it as fast as possible. We have had not 100%, but more than 95% success. Every once in a while, a fish dies, but for the most part, the surgeries, because they're so quick, that works. And also, if we do use radio tags on salmon, we just put them in their throat. We get them to swallow them, but because they die, and they're not feeding at the time, it's not going to interfere with their feeding. Whereas if you tried to do that with sheefish or some of the other fish, that would interfere with their feeding, and they're not dying. These fish live a long, long time. So yeah, just the surgeries are really, really quick and easy to do, and we've had really good success with that.
So we get, you had mentioned that you anesthetize the fish, you put the fish to sleep. What do you use to get them to go to sleep?
Our preferred method is it's clove oil, which you can buy from the store. We dilute it with some ethanol. It's been a long time remedy for babies' teething pain. It's a topical anesthetic, but because they're breathing it in through their gills, it puts them to sleep really fast. From putting them in the anesthesia to them reviving is usually less than 15 minutes. So there are other things out there, but that's the one that's mostly easiest to get into the field, because it doesn't require any special handling, and you don't have to have a license to have it like some of the other things that they might use in a hatchery or something like that. It's good stuff, at least in our opinion.
How can you tell when a fish is asleep?
They roll upside down. So one of the things that that a conscious and alert fish will do is it keeps himself upright in the water, where its top is sticking up and its bottom is down. And once they start losing that ability to stay upright, then and then we give them a little pinch on the fin, and it depends on how much they react to that pinch on the fin. If they pull back real quick, we'll let them sit in the anesthesia a little longer.
And is that the way that you figure out whether they've revived from the anesthesia as well? You pinch them and see if they react?
Well, as soon as we finish with the surgery, they go into fresh water in a holding pen, and somebody kind of sits there with them, keeping them upright. And then, especially if it's someplace that has good current, we'll face them into the current so they're getting fresh water over their gills. And at some point, they'll give a little twitch, and you kind of know that they're going to be able to stay upright. We have to be careful depending on what's around. We've had one fish, one of the smaller fish, the humpback whitefish. We let it go after we tagged it, and an eagle came down and swooped it up for dinner.
Actually, that connects to one of the questions that Chase in Mrs. Cudahy's class wanted to ask. He wanted to know, what happens if a predator eats the fish? Will the tracker track the new fish or animal?
Yes, it will, if it swallows it. So we had a problem with a whitefish in the upper Tanana River, which is close to the Canadian border on the eastern side of Alaska, and an eagle had caught the fish, not the same one that I've been talking about before, and took it up to feed its young. And because that transmitter was out of the water, its signal is really, really loud, and it made it difficult to hear other fish in the area that were muted by being underwater. So yeah, so it will track the fish. We knew exactly where that tag was. It was in the nest of this eagle. So
that's really funny. So one more question, because I know you have some other stuff to go on to, and we only have 20 minutes left. But how How would you suggest a middle school student gets involved in learning about tagging fish? You had mentioned the Tagging of Pelagic Predators website, which I'll send out to everybody for now, but are there other ways that a middle school student can find out more about tagging fish?
Well, if you have an agency that's near you, Fish and Wildlife Service or NOAA or Alaska Department of Fish and Game, or the state Fish and Game Department in your state, just contact them and tell them you're interested in fisheries and see how you can help.
Great, thank you so much. Oh, one more last question from Tuna Nick. What materials are tags made of?
So the radio tags there's a battery and then a circuit board that makes up the transmitter, and then they're all in epoxy resin. They just fill up a little container with that epoxy resin, and that's what the fish experiences is that resin. So it's a type of plastic, and it's very similar with the acoustic tags. And like I said, the T-bar tags are that same kind of plastic tag that you'd find on your clothing price tags.
Great, thank you. Well, I know that you have another section that has a lot of information, so maybe we should move ahead with that section.
Okay. So another quick question for you. What can we use to find out where the fish have been before we're able to tag them? So this is something that if you've been on this webinar before, they've talked about, but they've talked about it in a little bit different way. So what would the fish carry with it all the time that you might be able to use to figure out where the fish has been before you've captured it?
So this is a question for our listeners. What kind of thing could you use to find out where fishes have been before you could tag them? So where they were in the past, in other words. And Tunic is saying salt water in or on the fish, or what it ate. What it ate could maybe tell you where it's been. Grace is saying otoliths, and maybe we can talk about what otoliths are. Miss Doherty is saying their color of their skin. So that could be something like, I guess, if you're looking at salmon, they change color when they go into fresh water from salt water. They also, something else is where their predators were or their diet. Miss Kate's class is saying examine the parasites on the fish. So Bill, what do you think? What are you looking at?
Those are all good answers, and I'm sure you could find something from all of those different things. You could, yeah, we find sea lice that are only supposed to be in marine water on sheefish that are way upstream, and yeah, so parasites and what they've eaten and all those things. But what we use is otoliths. And otoliths can tell us a lot of things, not just about the fish that they come from, but even like the diet of marine mammals, because all otoliths from a particular species have a common shape to them. So you can see there are a whole bunch of different shapes here. There's probably, from my factory trawler days, there's probably some cod up there on the top and probably some rockfish on the bottom and maybe some flatfish in the middle. But what we do is we cut these otoliths and we get age, and then we do some microchemistry analysis. So depending on, so this picture that I just put up in the upper right there, we cut away everything from the bottom and the top, and it leaves us this slice in the middle. But depending on what you want to know and how old the fish are, you can cut these otoliths in a bunch of different ways to get the information that you need.
And Bill, could you explain for us what an otolith is again?
Oh yeah, sorry. So an otolith is an ear bone. So otolith is either Greek or Latin for ear stone. So they're hard bones. They're set between the eyes and the brain in the head of a fish, and all fish have them except for sharks and skates because they're not bony, so they don't have otoliths. But yeah, just like you have 3 ear bones, all fish have 3 ear bones. They just use them for different things. They use them more for balance.
Great,
So,
thank you.
oh yeah, sorry. So this is the oldest sheefish I've aged, and it came from the Selawik River, and I haven't even been able to find any scientific literature that has an older fish in it, so I think it's a record. Came from a big female, about over 4 feet long. So we count these rings just like we count the rings in a tree. So the first dot that comes up here is the number 1, is our first year, and then as they progress, they're in 5-year increments. So we'll quick count up the rings on this one.
That's a lot of rings.
Yeah. So this fish is 41 years old. So it's really old for a freshwater fish. There's many marine fish like rockfish and sablefish that live way longer than this, like 200 years. And there's a good otolith lab at the Alaska Department of Fish and Game has a list of all the oldest fish that their otolith lab has aged. But that's the first thing we'll do, is we want to know how old the fish is. And then the second thing we want to do is figure out where the fish have been. So we use 2 different instruments to do that, and they're really kind of complicated, and I've used one of them, and I am not 100% sure of how it works, so I don't expect you to learn this in 2 minutes. So the first one here is called an electron microprobe, and what it does is blast the otolith with an electron beam, and then we can tune the receivers on the sides to look for X-rays from particular elements. And there's a bit of a drawback with this, because the detectors can only detect certain types of elements, and they can only tell if the element was there or not. It doesn't really tell you very much about the type of element there or the isotope. So this is an otolith from my master's work. So in the picture on the left side is an optical image, what we'd see when we would, if we were counting the rings. And on the right side is what's called a strontium map, where we do the entire surface of the otolith to see the pattern of strontium in the otolith. And the graph on the bottom is a representation of if we took a line, we took a line and went right up where this division is, and this is the graph we get from that, and it's how much strontium was reflected at those particular points. And you can see that there's a big bright spot. Let me get back over here. There's a big bright spot right in the middle, and it starts from that little dark band there, and then goes to this. And we think that this is when it was down in the lower Yukon, like right almost in marine water. And you can see that there's that dark green line that's right above those arrows in the graph. That's our line. Everything below that is freshwater habitat, and everything above that is some type of either marine or brackish water. So you can see that this fish went above that line about 4 times, but spent the majority of its life before 3 years old and then after 8 years old in freshwater. So the second type instrument we use is called a laser ablation mass spectrometer. And I really don't know a whole lot about this. I know how it works, but what it does is it can look not only at a wider variety of elements, but it can actually look at different types of the same element. And in this case, we're looking at strontium-87 versus strontium-86. And those are isotopes of the same element, but one's heavier than the other. So we can take the strontium ratios, and this otolith here happens to be Chinook salmon. We can take those ratios and compare them to the same element or the same isotope ratio of water from a particular stream. And if those ratios match up along with some other elements so we can have corroborating evidence, we can tell exactly what stream this fish was raised in, where it reared in before it went to the ocean. And I've got one quick question for you. Can anybody give me an idea of why there's a peak in the core of this otolith?
So the core would be where the otolith would have started growing. Is that what it is?
So this is right. This is the center. This is the core here. And why this peak is right here? Because this is where it was when it was young. This is where its natal habitat, what the stream it grew up in. This is its isotope ratio. But why would it be high here when we know this is ocean habitat where it's really high here?
So Miss Ravenscroft's class is saying maybe it was in fresh water, although it seems like the habitat where it was a baby, which is the natal habitat, is really low there. So it almost feels like the core would be where it would be more close to salt water. So Michelle is saying maybe it was near the ocean when the otolith started to develop. So, and then Miss Doherty's class is saying maybe it's brackish water. So what's the answer there, Bill?
Those are all good guesses. So the core is always high because when the fish starts developing, it's using nutrients from the yolk, which means it's nutrients from where the female got its nutrients, which is in the ocean. So the core is marine nutrients, and then the natal stream is where it's absorbing nutrients and breathing, the water in fresh water, and then when it goes back out to the marine. So this section here is where the nutrients from the female came from. So that's why the core is always elevated.
That is very interesting, and I have to give kudos to Port Alexander because they said what you just said, just as you were saying it. They said it was maybe because the parent fish just came from saltwater, so they nailed it.
Yep, good job.
Yeah, and Ms. Doherty's class says one of our students, Isaac, said it's from the fish that it's from, the parents. So we've got some pretty smart kids online here.
Yep. Need a couple extra seconds of quiet time so they can get them. Okay. All right. So quickly, so this illustrates the difference between the two. So there I am. Come on. I got to get the cursor on the right screen here. There we go. So this fine line of dots here is what the microprobe makes. And those dots are 5 microns in size and they're 10 microns apart. So you can see, if you remember back to the how fine the lines were on that old sheefish, they would show you could get multiple dots inside each one of those lines when it's young, but where it gets out to the end, you're only going to get 1 dot maybe in those lines. But if you have the laser ablation, it just blasts a big, it's 25 microns wide, so it's very different scale of how precise you can tell which age this certain thing happened at. So they're two different instruments that give you similar data, but one's more precise and the other one's more versatile because you can look at a lot of different elements. So let's see if I can get off the page here. Okay. So now we got questions about otoliths. You can see up in the upper right-hand corner there, that's us taking a otolith out of a fish, out of a sheefish there. So.
All right. Well, we've got a couple minutes for questions. Actually, we got 4 more minutes in the webinar. So we did have a question from Ms. Ravenscroft's class about whether fish can live without otoliths.
No. No, we have to.
Otoliths for a lot of different things, don't they?
Yes. We have to take part of their head off to get their otoliths out. So no, they can't live.
Well, it's very interesting to me that you can find out so much about where the fish has been, whether it's been in saltwater or freshwater, especially for these species that move in between the two environments. It's interesting to see that you can find that out from chemistry on their otoliths. And so I think that we have only a couple more minutes left, so maybe we'll go to your next slide and just have your closing thoughts and see what you have, what kind of advice you have for our students.
Yeah, so I'll skip to the advice column. So if you, no matter what job you want to do, find something you really like doing. I hate doing paperwork. I hate analyzing data in the office, but I know every summer, fall, I get to go out and catch a bunch of big fish, and that makes up for all the time I have to spend crunching the data. As you know, somebody asked how they can get involved. You know, finding a mentor in your area. You know, it doesn't matter what you want to do. You know, find somebody who's also interested in those same things, whether it's, you know, science or computers or bookkeeping. It doesn't matter. You know, people always like to help people out with what they're passionate about. And then finally, you know, find something to work around problems. I had a terrible time with statistics. Statistics are super important to biological science because if you can't prove what you found with some statistical rigor, it doesn't mean much. But I had a terrible time getting through the advanced statistics that I had in, and I found some tools to get around those things, and a good tutor and a good instructor. So that's all I have.
Great. Well, thank you so much, Bill. And for everybody in our audience, I just sent out the emails for Story and Mabel and I. So if you have any other questions, I know we couldn't get to all of your questions, but if you have any other questions, or if you would like to find out more about educational resources we have or the Fish and Wildlife Service has, we can certainly forward them to you. And I think we are planning one more webinar in March, so please stay tuned, and we hope that we will be able to send you guys a flyer so that you'll be able to register for that webinar as well. So thank you, Bill, for all of your information and very interesting research that you've shared with us, and we are really happy to have you on as a presenter.
I really enjoyed it, and yeah, I'm always happy to talk about fish.
Great. Well, thank you, and thank you to everybody, and hope to see you the next time. Bye-bye.